रेगुलेशन ऑफ enzymatic activity. Enzymatic activity को रेगुलेट किस तरह कर सकते हैं? There are two general ways to control enzymatic activity. Control the amount or availability of the enzyme, control or regulate the enzyme's catalytic activity. Each topic can be subdivided into many different categories. Enzyme amount in a cell depend upon the rate in which it is synthesized and the rate it is degraded. Synthesis rates can be transcriptionally or translationally controlled. Degradation rates of protein are also controlled. However, we will be focusing on the regulation of the enzymatic activity. The catalytic activity of an enzyme can be altered either positively increasing activity or negatively decreasing activity through conformational alterations or structural covalent modifications. Examples already encountered is oxygen, carbon dioxide or BPG binding to hemoglobin. Also, substrate binding to the enzyme may also be modified by small molecule effect of changing its catalytic site. Protein phosphorylation of residues can active or deactive enzyme. These are generally hormonally controlled to ensure a concerted efforts on all tissues and cells. Aspirate, the first step in pyridine biosynthesis. This enzyme is controlled by allosteric regulation and feedback inhibition. Notice the S-shaped carving cooperative binding of aspirate. Positively homotropic cooperative binding CTP. Feedback inhibition, where the product of a metabolic pathway inhibits its own synthesis at the beginning or first committed step in the pathway. CTP is the product of this pathway and it is also a precursor for the synthesis of DNA and RNA nucleic acids. The rapid synthesis of DNA and or RNA depletes the CTP pole in the cell, causing CTP to be released from ATC and increasing its activity. When the activity of ATC is greater than the need for CTP concentration rises rapidly and rebinds to the enzyme to inhibit the activity. ATP activities, ATKs, purines and pyrimidines are needed in equal amount. When ATP concentration are greater than CTP, ATP binds to ATCase, activating the enzyme until the level of ATP and CTP are about the same. Enzymatic catalysis and mechanism. Number A, acid-base catalysis. B, covalent catalysis. C, metal-ion aided catalysis. D, electrostatic interactions. E, orientation and proximity effects. F, transition state binding. Journal acid-base, rate increased by partial proton abstraction by a prostate base or rate increased by partial proton donation by a prostate